good afternoon, everyone. My name is Samuel Fuentes. I conducted my honors thesis in the CRAMS lab, and in my time there, I learned a great deal. I'm really excited to share with you some of my findings. So let me give you some context into my study. We are interested in the relationship between the immune system and organ transplantation. Now, I'm sure you've heard a lot about the immune system in the last few years, and that's because we typically think of it as protecting us against pathogens such as viruses and bacteria. However, this fact presents some interesting complications in organ transplantation. Here, an example of a kidney transplant where when we transplant the organ from a foreign tissue, the immune system recognizes that tissue as foreign and begins to initiate inflammation and the cells attack that graft in immune response. Now, our goal in transplant immunology is to understand the immune response on a deeper level to improve this therapy for the patients who need it. Now let's group the immune system into two big buckets. That's the innate immunity and the adaptive immunity. Most of what we know about organ transplantation is concerning this adaptive immunity, the lymphoid cells, T cells, and B cells. However, I was interested in the innate immunity with these myeloid cells, macrophages and granulocytes and monocytes. The reason for this is because these are the first cells to respond to an immune reaction. They're fast-acting, and a really important role is that they activate and teach the adaptive immunity about the target that needs to be attacked. So for the rest of this talk, I want to focus on the relationship between these myeloid cells and this type of transplant, a vascularized composite tissue allograft, or VCA for short. So VCA is a relatively new type of transplant where, like you can see here in this limb transplant, we're transplanting multiple types of tissue at once: muscle, skin, fat, nerves, and this provides multiple sources of antigen for our myeloid cells to interact with, and also multiple sites of inflammation. And what we've seen clinically is that these type of transplants have very strong rejection in patients, even with heavy immunosuppressants. And also, there are a few studies investigating the cellular and molecular pathway of rejection in VCA, and none at all investigating how the myeloid cells play into this. So given all this led us to the question, what are the characteristics of the myeloid population in rejecting VCA? Understanding these initial early reactors and activators of the immune system might help us understand rejection overall, and of course lead to better outcomes for this type of transplant. So how do we go about answering this question? Well, our in-lab microsurgeon created a limb transplant model in mice. So we grafted a limb from one mouse onto a recipient mouse. They were grafted on, the circulation was connected fully, and so this was a heterotopic transplant, so the limb wasn't functional, but it still allows us to observe the immune response that's going on. It's a very common practice in transplant immunology. We had two important models. The first is a syngeneic model where the donor limb and the recipient were genetically identical, so there was no rejection at all. We then had an allogeneic model where there were several differences between our limb and our transplanted and our recipients. So this rejection, this mismatch we observed several signs of rejection in this mouse. Comparing this rejecting versus non-rejecting model then will allow us to see what's going on in the immune system. So like I said, we isolated the myeloid cells and we used mass cytometry to observe the protein expression on these cells. Now, protein expression is really interesting because this allows us to see what type of cells we're looking at and also observe their activation state and their function, what they're doing. We used a panel of 45 protein markers, and with that we had to perform dimensionality reduction and clustering to find our findings and pull our information out. So here's an example of that dimensionality reduction. Each dot here is a cell, and like I said, we had 45 proteins that we're measuring. We can't create a 45 dimension plot, so we use UMAP to bring these cells down, this data down into 2 dimensions and represent each dot so that it's grouped together based on how similar it is based on that 45 proteins that we're expressing. So now we're able to identify what type of cells we're looking at. And here in the blue we have our granulocytes, down here we have our monocytes and macrophages, and in light blue dendritic cells. And we were able to separate our allogeneic and syngeneic models, so now we can make those comparisons. So one example of the differences we found was that there is an increase in monocytes and granulocytes in our allogeneic, in our rejecting group. Monocytes are a really interesting cell. They differentiate into dendritic cells and macrophages, and granulocytes release out granules, or like digestive proteins, and also engulf other cells and other bodies, digesting them as well. So we have this difference in proportion of cells in our rejecting group, but what are they expressing? What's on these cells, and which tells us what they're doing? So this is that same UMAP, except now we're looking at MHC2 expression. So in yellow and green, that represents high MHC2 expression, and in blue and purple, that's low MHC2 expression. So in the monocytes, it's really interesting that we saw that the monocytes were responding to alloantigen in our allogeneic group and upregulating MHC2, compared to our syngeneic model, which has much less expression, as you can see. MHC2 is a really important molecule in transplantation because this is the molecule that helps teach the adaptive immunity cells what it is they're targeting against. So we see that our monocytes, again, in our allogeneic group are upregulating that. Now, we had a more moderate increase in granulocytes in our allogeneic group, but what was interesting and surprising was that there was a small subpopulation that were expressing PD-L1. Now, this was an interesting finding, and we suspect that this is because these PD-L1 positive granulocytes were participating in the resolution of inflammation, the immune response, or perhaps just responding to general inflammation. Also, we found that there was a population of granulocytes that were positive for YAY. Now, YAY is a marker that allows us to see which cells are positive for donor limb antigen. And this is interesting because cells that are positive for donor limb antigen, such as these granulocytes, we're interacting with the donor limb, we're interacting with that material in some way, uptook it into themselves, and are now displaying it on their surface. And again, this is going to help teach the adaptive immunity what type of target they're looking for, and then proceed to let them attack it. So overall, I hope I've impressed on you that these two cells are having some sort of pronounced role in VCA rejection. In conclusion, we conducted this study where there was no information before, so all these findings are pointing us in the direction that will hopefully allow us to conduct further studies to understand what's going on cellularly and molecularly. Of interest are the monocytes and granulocyte population, and so we can conduct further studies to test exactly what is the function of these cells, and what are they, if we take them out, what is happening in the graft, or if we enhance them, what is happening as well. Multiple studies in power. Also, because myeloid cells are these early immune activators and reactors, we can validate some of these findings to serve as early biomarkers of rejection. This is important, of course, clinically, and also important because we can start to refocus some of our therapeutics to perhaps include, to improve VCA for patients, such as this patient here, who was the first pediatric bilateral limb transplant. And before I take questions, I'd like to thank my family, my mentors and friends in lab, Sherry Krams, James, and Gianna, and then the people who came to support me today. Thank you all.